Hey y'all, my name is Kayla Hurst, and I'm going to be presenting on genome editing and the CRISPR-Cas9 technology. First, I want to go over today's agenda. I'm going to first touch on the background and key terms that will be used throughout the presentation, then dive right into CRISPR and Cas9 technology. I'll be discussing how it works, their therapeutic approaches, the future of CRISPR, ethical concerns that were discovered, and at the end, close with discussion questions. In the body, there are multiple cells containing copy of genomes, which consists of over 20,000 genes and 3 billion letters of DNA. DNA consists of separate two strands twisted into a double helix and held together by a pairing rule. A pairs with T and G pairs with C. These genes have an effect on our health, many of which have been identified to affect our risk of getting diseases. Researchers have been able to identify new technology that can edit and repair our DNA that contains some of those diseases. CRISPR technology is a simple and very powerful tool to edit genomes. Research have been able to easily alter DNA sequences and modify the genetic function. It has many potential applications, including the correction of genetic defects, treating and preventing the spread of diseases. Its popular usage is CRISPR. It is shorthand for CRISPR-Cas9. The protein Cas9 is an enzyme that acts like a pair of molecular scissors capable of cutting the strands of DNA. So, how does CRISPR-Cas9 edit genes? They edit genes by precisely cutting the DNA and allowing natural DNA repair process to continue. The Cas9 enzyme acts like molecular scissors to cut that DNA at the location specified by a guide RNA. There are three main categories of repairing DNA that can be performed with the CRISPR-Cas9 technology. It can either disrupt, delete, or correct and insert. The CRISPR-Cas9 technology disrupts RNA when a single cut is made. A process called non-homologous end joining can result in an addition or deletion of the base pairs, disrupting the original DNA sequence and causing gene inactivation. When CRISPR-Cas9 deletes part of the DNA, a larger fragment of DNA can be deleted by using two guide RNAs that target two separate sites. The non-homologous end joining unites the separate ends, deleting the intervening sequence altogether. Lastly, CRISPR-Cas9 can correct or insert our DNA. By adding a DNA template alongside the CRISPR-Cas9 machinery, allows the cell to correct a gene or insert a new gene altogether. This process is called homology direct repair. So I wanted to talk about CRISPR therapeutics. The mission statement states that at CRISPR Therapeutics, they are focused on developing transformative gene-based medicines for serious human diseases. CRISPR Therapeutics is rapidly translating specific, efficient, and versatile CRISPR-Cas9 gene editing platform into therapies to treat cancer, diabetes, and other diseases. CRISPR-Cas9 aims to target the source of genetically defined diseases and engineer the next generation of cellular therapies. Genetically defined diseases have genetic cause, including more than 10,000 monogenic disease caused by mutations in individual genes. CRISPR-Cas9 has the ability to repair the genetic defects that underlie these diseases. Cellular engineering are cell therapies to make a meaningful impact in certain diseases. CRISPR-Cas9 can engineer the genomes of cellular therapies to make them successful, effective, safer, and available to a broader group of patients, with both therapies that can edit cells, either ex vivo, which is outside the body, or in vivo, which is inside the body. So to better understand the CRISPR process, so as mentioned on the previous slide, there are two ways that these therapies edit cells. The first is ex vivo, which removes cells from the body, and then CRISPR-Cas9 is delivered to the cells in a culture to produce the desired edit, and then the edited cells are then returned back to the patient. For in vivo, the CRISPR-Cas9 components are packaged in a delivery vehicle such as a lipid nanoparticles that are safely and effectively deliver nucleic acids, overcoming a major barrier and preventing the development and use of genetic medicines. Then the therapeutic is delivered systematically to a target organ such as the liver. So now that we've kind of talked about CRISPR-Cas9 technology, I wanted to use this slide to kind of take us through the progression and kind of explain how it works. So as you see at the first point, we have Susan who has sickle cell disease caused by a single DNA letter mutation. So then you can see that DNA double helix with the sickle cell mutation. Then at step 2, in the lab, scientists create a strand of guide RNA that matches the mutated DNA sequence. In step 3 is when Cas9 comes into play. Cas9, or also known as the molecular scissors, is added to the RNA mix. Step 4, Susan receives an injection of that RNA Cas9 mix. Then, guide RNA identifies that mutated DNA that causes the sickle cell disease. Then step 6, Cas9 cuts out that affected sequence. In step 7, scientists insert a healthy DNA sequence to replace the clipped sequence that we just cut using Cas9. Lastly, if all goes smoothly, Susan will no longer suffer from sickle cell disease and will have a newly healthy DNA strand. So going off what we've learned thus far, the future of CRISPR-Cas9 has a lot in store for us. In the future, repairing DNA will continue to transform not only healthcare, but our humanity as a whole. The way CRISPR is moving, individuals will be able to enhance their health by the ability to have their DNA repaired. This includes the end to diseases including HIV, sickle cell anemia, AIDS, Down syndrome, and many more. Not only disease, but it could be a cure for cancer as well. CRISPR can improve by being able to detect the multiplying cancer cells in the human body more precisely and edit them throughout our DNA. Another way DNA can be modified is through designer babies. The idea being, if we're able to repair and modify DNA to eliminate a certain aspect we don't like, maybe then we can do the reversal as well. Being able to choose for your child to not have certain genetics passed on to them. In the previous slide, I mentioned designer babies. So I want to go into a little bit of ethical concerns that are around this topic. There are a lot of ethical concerns around the fact of designer babies. The means to edit the genome of an embryo exists. There are different personal opinions on the genetic engineering of an embryo. Regardless of those opinions, it will still affect everyone in some way. If humans begin to be modified, it can change the genome of the entire species as a whole. Though this would take a lot of time to do, it would be due to their engineered traits being passed down to their children. It's thought that it would first eliminate those DNA that will cause a child to eventually have Alzheimer's, or if they have sickle cell anemia. But if that could be fixed, why not give them perfect eyesight, or the perfect height or muscle mass? Or if you want your child to have blonde hair and blue eyes. The last thing I wanted to cover was the financial impact, in particular on healthcare. The utilization and the advancement of DNA repairing in regard to genetic disease or cancer will decrease, ultimately reducing the amount of services and dollars spent on those services the patients may need. Depending on how individuals will pay for these services will be another topic on how it will impact a healthcare system financially. I want to move on to insurance companies. Assuming insurance companies will begin to be involved in covering DNA repairing, the type of repair and the category it would fall under will impact the finances differently as well. Insurance companies will have to determine how much they are willing to cover and what they will cover. For example, how much will they cover of a sickle cell anemia change versus I want my baby to have blue eye change? Another financial healthcare impact will be centered around regulations, ensuring they are FDA approved, regulated, and tested. With regulation comes on the concept and concern of the black market. If it is not regulated and tested, what is the profit and safety concerns that we are facing? Repairing DNA is not way off in the future. It is starting to happen now and will continue to take a larger presence in our healthcare system. I wanted to go over some discussion questions on CRISPR-Cas9 that I've been thinking about and have also mentioned throughout the presentation, but really want to hear your input. We talked about the ethical concern of designer babies, but what are some other concerns or challenges that should be considered when introducing CRISPR-Cas9? I also talked about the financial impact on our healthcare system in particular, but I want to know your thoughts on that and maybe just the impact on finances overall, not just in the healthcare system. Going along with that, I did mention a little bit about insurance companies. I personally am not in the insurance business or world, and so if any of you are, I'm interested to see if you guys had any more thoughts on that. And lastly, what other technology can be used alongside with CRISPR? And what I mean by that is we've learned about a lot of different technology this semester. I personally wrote something, I think, in one of our discussion boards about 3D printing. And that's maybe something that CRISPR and 3D printing can kind of use together to even make, cut down on diseases or helping with organs better as well. So I just wanted to hear your guys' thoughts on that. I really appreciate you listening to the presentation. I hoped you learned something new today, and I can't wait to read your comments. Thank you.